Welcome back to the deep dive. Today, we're tackling maybe one of our most complex topics yet. We're really plunging deep into the visual system.
Yeah, it's huge.
We aren't just looking at, the eye's anatomy. We're digging into that whole layered process, how reflected light becomes the reality you actually experience.
Exactly. And it really is a kind of illusion, isn't it? Because vision doesn't just happen in the eyes. That's just the start.
Right.
It's the eyes capturing data, the optic nerves translating that into electrical signals, and then the brain working like crazy to process, predict, and ultimately build that reality.
So our scope today is pretty broad then.
It has to be. We're looking at the biology, this elegant but frankly flawed machinery. Then the brain's processing pathways, which are incredibly sophisticated, and importantly, the limits of that system.
Limits that we're now pushing up against with technology, AI, bioengineering.
Precisely. All that.
Okay, and the sources we've pulled together reflect that breadth. We've got stuff from, well, pure ophthalmology, connecting how the retina works to, believe it or not, Monet and art history.
That's a fascinating link.
Then there's some really high-stakes ethical reporting on bionic eye implants.
That's crucial context.
And of course, cutting-edge neuroscience out of MIT, that's actually questioning some really long-held ideas about how the brain handles vision.
Yeah, challenging the old models. It's exciting stuff.
So the mission here is to pull out the key insights from all this, the really critical nuggets.
Right, so that by the end, you, listener, hopefully walk away with a fundamentally new way of thinking about how you see the world, why it looks the way it does.
Okay, let's unpack this. And I guess we have to start right at the beginning. The eye itself, often called a masterpiece, but our sources say maybe not quite. Deeply flawed, actually.
Flawed, yes, but maybe masterpiece of evolution is fairer. Because evolution isn't perfect engineering, it's adaptation. The eye is an exquisite instrument, biologically speaking, but it's fundamentally an adaptive construct. It's been shaped by countless trade-offs over millions of years.
Prioritizing survival over, perfect picture quality.
Exactly. It's not an infallible machine for capturing objective reality. It's a good enough system for keeping you alive in your environment.
And one of the biggest flaws comes from how it's wired up, right? This backwards design people talk about. What does that mean exactly?
It sounds completely counterintuitive, but yeah, our photoreceptors, the rods and cones that actually capture the light photons, they're oriented away from the incoming light.
Wait, so the light has to go through other stuff first?
Yes. It has to pass through the whole neural network of the retina, all the processing cells, before it hits the light-absorbing pigments, which are tucked away at the very back of the photoreceptor cell.
That seems inefficient.
It is. And because of that orientation, all the nerve fibers carrying the visual signals away from those photoreceptors have to run across the front of the retina.
Okay, I think I see where this is going. They all have to bundle together somewhere to leave the eye.
Precisely. They all have to exit the eye to form the optic nerve, which goes to the brain. They converge at one single point called the optic disc.
And if that spot is full of exiting nerves?
There's absolutely no room for any photoreceptors. No rods, no cones, nothing to capture light there.
So functionally, it's a blind spot.
A literal hole in your visual field.
But we don't notice it, do we? I mean, I'm not aware of a gap in my vision right now.
Almost never. Your brain is incredibly good at perceptual filling in. It uses the information from the surrounding areas, plus prediction based on past experience, memory.
To just seamlessly patch over that hole.
Exactly. Imagine how disorienting it would be if you consciously registered that blind spot all the time.
Yeah.
It would be a nightmare for just, walking around.
So this simple evolutionary quirk, this design flaw, forces the brain into doing constant active prediction just to give us a stable, unified view.
That's a key part of it, yes. And what's really fascinating is comparing this to other animals. This flaw isn't like a universal law of eyes.
Oh, who got it right?
Well, right is relative, but look at cephalopods. Squid, octopus. They evolved these large, complex, lensed eyes independently from us vertebrates. It's a classic case of convergent evolution.
They look kind of similar on the outside.
Remarkably similar.
Yeah.
But their photoreceptors are oriented the logical way toward the incoming light.
So they're nerves.
Their visual nerves run behind the retina. They go straight out the back of the eye. They don't need to cross the light path at all.
Meaning no blind spot.
No blind spot. It really drives home that the way our eyes are built is a result of our specific evolutionary history, full of contingencies. It wasn't the only possible solution.
Okay, so we have this built-in flaw. But despite that, primates really lucked out in another area, right? Color vision.
We absolutely did. Our retina is what's called a duplex retina. We have rods, which are super sensitive for seeing in low light, black and white, basically. And then we have cones for daylight vision, which give us color and high detail.
And most mammals don't have the same setup.
Most mammals are dichromatic. They only have two types of cone cells, usually sensitive to the blue and maybe greenish parts of the spectrum. But somewhere along the line, primates evolved the third type,
The red one?
the L-type cone, yeah. Sensitive to longer wavelengths, what we perceive as red.
And having three cones, L, M, and S for long, medium, short wavelengths, that's the key to our rich color world.
That's the foundation. Red, green, blue sensitivity. Having those three channels allows the brain to compare the intensity signals from each type.
And figure out the exact colors.
By calculating the relative differences, yeah. It lets us distinguish, well, estimates vary, but up to maybe 10 million different colors.
Wow. Compared to most other mammals, that's...
Yeah. It's a massive upgrade in visual data. Huge advantages, like, as you mentioned, spotting ripe red fruit against green leaves, or subtle changes in skin tone, potentially important for social cues.
Okay, so that color resolution, and really all our vision, depends on those signals getting from the eye to the brain accurately and crucially synchronized.
Absolutely.
And this next detail from the sources about how the eye compensates for physical distance within itself. It's just mind-blowing. It feels like biology correcting its own geometry problem.
This is really elegant. It involves the retinal ganglion cells, the RGCs. These are the output neurons of the retina. Their axons form the optic nerve. They're the only pathway carrying visual info to the brain.
Okay.
Now think about the layout. The RGCs near the center of your vision, the fovea, especially those on the side towards your temple, the temporal side, they have the longest way to travel to reach the optic disc where they exit the eye.
Right, compared to the ones on the nose side, nasal side, which are much closer to the exit point.
Exactly. So logically, the signals from those far-away central RGCs should take longer to arrive at the optic disc than signals from the closer peripheral RGCs on the nasal side.
And if that happened, our vision would be a mess, wouldn't it? The center lagging behind the edges, a totally disjointed image over time.
It would be completely disorganized. Temporal dispersion, it's called. The brain needs those signals, especially the ones triggered at the same instant by something in the visual field, to arrive at the visual cortex pretty much simultaneously to build a coherent picture.
So how does the eye solve this timing problem?
This is the amazing part. Researchers found that the RGC axons that have the longest path to travel those ones in the temporal region actually propagate their electrical signals, their action potentials, significantly faster.
Faster? How much faster?
More than 40% faster than the axons that have a shorter path, like those in the nasal region.
Whoa! So the longer the wire, the faster the signal. That's the opposite of how electrical cables usually work.
It's active biological compensation. It's like there's a built-in speed adjustment based on distance. The axon's properties, perhaps its diameter or myelination, are tuned so that the longer physical distance is offset by faster signal transmission speed.
To ensure they all arrive at the finish line, the optic disc, at roughly the same time.
Exactly. This correlation between the axon length and its signal speed actively reduces that potential temporal dispersion. The measurements show the difference in arrival times for signals triggered simultaneously across the retina is squeezed down to less than 2.5 milliseconds.
2.5 milliseconds. That's incredibly precise synchronization.
It has to be. It ensures that what you see in your central vision and what you see in your peripheral vision are temporally locked together, maintaining that smooth, unified visual field we take for granted.
That's genuinely incredible biological engineering. If that system failed, say, due to a disease affecting those longer axons.
You could definitely experience problems. Temporal desynchrony. Maybe not obvious jitteriness, but perhaps issues processing fast motion, or a subtle blurring or smearing, especially comparing center and periphery. That tiny 2.5 millisecond window shows just how critical this timing is.
Okay, that initial speed fix is seriously complex. Now let's move beyond the RGCs and their wiring, into the brain's first big decision about the signals arriving, particularly this idea of color versus brightness.
Right, and this brings in that ophthalmology perspective, which often looks at the evolutionary history. The insight here is that our ability to see all those colors, the 10 million hues, that evolved relatively late.
After the more basic stuff was already in place?
Long after the fundamental retinal circuitry for detecting contrast, edges, shapes, depth, motion, all that stuff was established. And those older, arguably more critical systems rely primarily, sometimes only, on differences in brightness or luminance.
So color is almost like an overlay, an extra layer of information added onto a system built for black and white contrast.
In evolutionary terms, yes, you could think of it that way. It's superimposed. And this explains why brightness, or value as artists call it, generally trumps color when your brain is trying to figure out the shape or form of something.
That makes sense. You can recognize a black and white photo instantly, even though all the color info is gone. The shapes are still clear.
Exactly, and artists know this intuitively. You can paint a scene with totally unrealistic colors, a purple sky, orange grass, whatever, but if you get the light-dark contrast right, the image still looks solid, recognizable, three-dimensional.
And the clearest scientific demonstration of this brightness first principle is that phenomenon called equiluminance.
Ah, yes, equiluminance. It's a fantastic visual trick. It happens when you have two different colors, say red and green, that happen to have the exact same physical brightness, the same luminance
So
level.
different hue, same intensity.
Precisely. Now, remember those older visual pathways, the ones crucial for detecting form, edges, location? They rely heavily on luminance contrast. So when you present them with an image where there is no luminance contrast,
They get confused.
profoundly confused, they struggle to find the boundary. If you put, say, green letters on a red background and they are perfectly equiluminant, those letters become incredibly difficult to read. They seem to shimmer or vibrate, the edges become unstable.
Because the form detection system is starved of its primary cue, the light-dark difference.
Exactly. And if you were to convert that equiluminant red and green image to grayscale, what would you see?
Just a flat, uniform, gray field, right. The boundary would completely disappear.
Completely. It shows how much the brain leans on brightness changes to define borders.
Yes.
Without that contrast, shape and location become ambiguous.
And you mentioned artists exploiting this, Monet.
Claude Monet is a master of this. Look at his painting Sunrise Marine. Those famous orange-pink reflections of the sun on the water. They're painted with colors that are very close in brightness, nearly equiluminant to the surrounding grayish-blue water.
Okay. So what effect does that create?
Because the brain's form systems can't quite lock onto a sharp, stable edge for those reflections, they don't look like solid patches of paint. They seem to shimmer, to glow, almost to move independently on the water's surface. It creates that incredible sense of flicker and light.
Whereas if you'd use, say, bright white paint for the reflections,
That would create high contrast. It would look much more static, solid, less alive. Monet uses the instability inherent in near equiluminance to capture the feeling of dawn light on moving water. It's genius.
that's fascinating. Contrast that with someone like Kirchner, the German expressionist. He seems to do the opposite.
Ernst Ludwig Kirchner, yeah. In many of his drawings or woodcuts, you see these bold, sharp, black outlines defining buildings, trees, figures. But the color inside those outlines is often, well, wildly inaccurate. Splotches of color bleeding outside the lines or just the wrong color altogether.
But you can still tell exactly what it is.
Instantly recognizable, isn't it?
Right.
Why? Because those high-contrast black outlines provide such a strong signal to the fundamental form and depth perception systems.
The brightness difference is huge.
Massive. Once those boundaries are powerfully defined by the luminance contrast of the black lines, the brain essentially says, okay, I know what shape this is, and it almost doesn't care that the color information inside is messy or wrong. It fills it in, constructs the form based on the dominant contrast signal.
So again, contrast defines the shape. Color is secondary detail.
It's a powerful demonstration of that hierarchy in visual processing.
Okay, so our perception can be tricked by things like equiluminance, but generally, in the real world, our brain does an amazing job of keeping colors stable, right?
Yeah.
Even when the lighting changes dramatically. That's color constancy.
Color constancy, yes. It's one of the brain's real superpowers for navigating the world. Think about it. The light actually hitting your retina, the optical image, depends on two things interacting.
The object itself and the light shining on it.
Right. The object's intrinsic surface properties, what makes banana skin reflect yellow light, and the extrinsic illumination is a yellowish sunlight, bluish shade, greenish fluorescent light.
So if the light source changes color, the light bouncing off the banana and hitting my eye should also change color quite a bit.
Logically, yes. The raw data changes radically. Yet, you still perceive the banana as yellow, pretty much regardless of whether you see it outdoors in sunlight, indoors under a warm incandescent bulb, or under a cool LED.
My brain is somehow subtracting the color of the light source.
It's actively calculating and compensating for the illumination in real time, trying to estimate the true surface reflectance, the object's inherent color. It's a complex computational feat.
And how good are we at it? Did the study show it works well?
Remarkably well, especially in natural settings. Researchers test this by having people match colors, often using standardized color swatches called Munsell chips under different types of controlled lighting. They measure the errors using a standard unit called delta E.
Okay, delta E, what does that actually measure? How big is a unit of color difference?
Good question. Delta E is the standard way. Color science quantifies the perceptual difference between two colors. How different do they look to a human eye? A delta E of less than 1 is generally considered an invisible difference. You wouldn't be able to tell them apart.
And the errors in these color constancy tests,
They found the mean error was only about 5.5 delta E units.
which sounds small.
It is small. It's a noticeable difference if you have two colors side by side, but it's small enough that it's likely within the range of slight inaccuracies in just remembering a specific color. Over time, our long-term color memory isn't perfect either. It suggests the constancy mechanism itself is working really well.
Not a massive failure to adjust, just minor variations.
Exactly. And what's more, the studies found something really interesting. Our color constancy is better for the kinds of illumination changes we typically encounter in nature.
Like
Yeah.
the shift from sunlight to cloudy daylight.
Precisely. Changes along the yellowish-bluish or greenish axis, typical daylight variations. Our constancy mechanism is more accurate for those ecologically relevant shifts than for more unusual artificial lighting changes, like deep reds or violets.
So our brain is evolutionarily tuned to subtract the lighting variations common in our natural world.
It seems highly optimized for the environments where it evolved.
Okay, that whole process, figuring out intrinsic color from extrinsic light, that requires some serious high-level computation in the brain, right? In the cerebral cortex.
Absolutely. This is where perception really gets constructed.
And for decades, the standard model for how this happened was the dual stream hypothesis.
Yes, that was the dominant idea, really established back in the 80s. The thinking was that once the basic visual information gets processed in the primary visual cortex, V1, it splits off into two major pathways heading towards higher cortical areas.
Right, you had the ventral stream running down the temporal lobe, often called the what pathway.
Its job was thought to be mainly object recognition, identification, figuring out what you're looking at. Is it a face, a tool, an animal?
And then you had the dorsal stream heading up towards the parietal lobe,
Yeah.
the where pathway.
Or sometimes the how pathway, responsible for spatial stuff. Where is the object located? How far away is it? What's its orientation? And crucially, guiding your actions based on that spatial information, like reaching out to grab something.
Seemed like a pretty neat division of labor. What versus where?
It was a very influential model. Clean, logical, but like many clean, logical models in biology,
It's getting complicated.
is being seriously challenged, yes.
Yeah.
Particularly by some recent work using deep learning models coming out of MIT.
The AI angle again. How are they using AI to question this fundamental brain map?
Well, they used artificial neural networks, specifically convolutional neural networks, CNNs, which are sort of inspired by the hierarchical structure of the visual system itself. They trained these models in two different ways.
Okay, what were the two training regimes?
One set of models was trained, basically, on the classic ventral stream task, object categorization. Learn to tell cats from dogs, cars from bicycles, what things are.
Makes sense. You'd expect that model to end up looking something like the brain's ventral stream.
Right. But the second set of models was trained purely on spatial tasks. Things like figuring out an object's exact 3D location, its orientation, its size relative to other things, purely where information.
Okay, so you'd expect that model to maybe align better with the dorsal stream, the where pathway in the brain.
That was the prediction based on the classic dual stream hypothesis. But here's the kicker.
What did they find?
What's fascinating here is the models, trained only on those spatial where tasks, turned out to be surprisingly good predictors of the neural activity actually measured in the brain's ventral stream, the supposed what pathway.
Wait, really? The where-trained AI matched the what part of the brain?
They showed what the researchers called strong neural alignment. The internal workings of the AI, trained to figure out where things are, ended up resembling the brain activity patterns in the area we thought was mostly dedicated to figuring out what things are.
That seems to really blur the lines between what and where.
It absolutely does. It strongly suggests that the ventral stream isn't just narrowly focused on object labels. Its core function might be much more integrated, perhaps deeply involved in processing complex spatial information as a foundation for recognition.
Like, if you fully understand the 3D shape and location of all the parts of an object.
Then figuring out what it is becomes much easier.
Right.
It suggests the optimization might be for something more fundamental, like understanding scene geometry, which then supports categorization. It really throws the primary function of this huge chunk of visual cortex wide open for debate again.
That's a major shift in thinking. So, getting that full picture requires processing through multiple stages in the cortex, doesn't it? Let's follow the information flow deeper.
Okay, so after the initial processing in the retina and the thalamus, the signal hits V1, the primary visual cortex. This is like the first main sorting office.
What does V1 figure out?
Basic, fundamental features. It's meticulously mapped for things like detecting edges and their orientation, those famous orientation columns discovered by Hubel and Wiesel. And critically, it's where the signals from the two eyes first get compared to start calculating depth from binocular disparity.
The difference between the left and right eye views.
Exactly. V1 does that initial comparison, then the information moves on to V2.
And V2's role, you mentioned it helps with color constancy earlier.
Yes, V2 acts as a crucial processing step. It helps stabilize our perception by starting to disentangle the object's true color from the color of the ambient light shining on it. Structurally, V2 has these distinct zones, like thin stripes, where color-selective neurons are concentrated, highlighting its role.
Okay, so V1 does edges and basic depth. V2 works on color stability. What about V4 and IK? These sound like higher level areas.
They definitely are. V4 seems to get involved in gauging depth intervals, not just is A in front of B, but how much space is between A and B. It deals with relative disparities and contributes to more sophisticated color constancy, building more stable representations of objects regardless of viewpoint or lighting.
An IT inferior temporal cortex. That sounds like the top level for object recognition.
IT is considered a real powerhouse for representing complex 3D surfaces and overall object shape. It integrates all the features processed in earlier areas into coherent object representations.
And this is the area where AI models trained on its activity become more robust.
That's right. When AI models are optimized to mimic the neural activity patterns found in IT cortex, they show improvements in things like robustness to image variations and, interestingly, become more resistant to those adversarial attacks.
Those weird, subtle image changes that can completely fool an AI but are invisible to humans.
Exactly. Training AI based on IT activity seems to confer some of that human-like perceptual stability and robustness. It suggests that IT's way of representing objects is particularly effective and perhaps less brittle than simpler feature combinations.
Okay, so that complex 3D reality built up through V1, V2, V4, IT, that's crucial for survival. Let's focus now on the dynamics, how the brain calculates depth, how it wires itself up, and how it handles a moving world.
Right, depth perception itself.
Hmm.
It's hard to overstate how critical this is. Without it, the world would just be a flat, confusing mess. You couldn't navigate, you couldn't interact with objects properly.
And the main cue is binocular disparity, using the two eyes.
It's the most important cue for fine depth judgments, yes. That difference in the image seen by the left and right eye, caused by the horizontal separation of our eyes on our head. The brain is constantly calculating that disparity to figure out how far away things are.
And that calculation isn't done all at once. It's staged across those cortical areas we just talked about.
Exactly. Early areas like V2 figure the basics, which surface is in front, which edge belongs to which object, border ownership. Then intermediate stages, like V4, seem to gauge the actual depth intervals, the relative distances.
And IT pulls it all together into a 3D shape.
IT represents the more complex global 3D surface geometry, although interestingly, some research suggests that even this high-level representation in IT might still be somewhat viewer-centered.
Meaning tied to my current viewpoint rather than a totally objective, abstract 3D model.
Potentially, yes. It might represent the object's shape, as seen from here, which is arguably more useful for immediate interaction than a purely abstract model. Still an area of active research.
And the brain isn't just born with all these connections perfectly wired for depth perception, is it? The sources talked about intense rewiring, plasticity, especially early on.
Oh, absolutely. The visual system isn't rigidly hardwired from birth. It undergoes a profound period of refinement, particularly during what's called the critical period in early development. Experience shapes the circuits.
And this isn't just minor tweaking, right? The study on mice developing binocular vision sounded pretty dramatic.
It really was. They tracked individual synaptic connections, the little spines on dendrites, where connections happen over time in the visual cortex of young mice as they were first using both eyes together. What they saw wasn't just strengthening or weakening existing connections, they saw massive, extensive rewiring.
Like connections being constantly added and removed.
Constantly. It sounds chaotic, but it's a highly dynamic process of selection. Over just 10 days during this critical period, they found that only about 40% of the synaptic spines that were there at the beginning actually survived.
Wow, 60% were eliminated or replaced.
Yes, it's this intense turnover, continuous addition and removal of synapses that ultimately sculpts the final, refined, and stable circuit needed for precise binocular vision.
What drives that selection process? How does the brain know which connections to keep and which to prune?
It seems to be driven by fundamental principles of neuroplasticity, primarily activity-dependent mechanisms. Two key ones are often mentioned: Hebbian plasticity and heterosynaptic plasticity.
Okay, Hebbian is the fire together or wire together idea.
Basically, yes. Synapses that are consistently active, meaning the presynaptic neuron successfully causes the postsynaptic neuron to fire, get strengthened and stabilized. If a connection is part of a circuit that's effectively processing relevant visual information, like detecting lines of a specific orientation from both eyes, it gets reinforced.
And heterosynaptic plasticity, what does that do?
That relates more to the competition and correlation between neighboring synapses. It can mean that synapses that are weak, inactive, or whose activity doesn't correlate well with the stronger, more successful neighbors tend to get weakened or actively eliminated. It's the pruning mechanism.
So it's use it and correlate or lose it.
Pretty much. Together, these mechanisms ensure that the connections which are most useful and reliable for processing the specific patterns of visual input the animal is experiencing are the ones that survive and form the mature visual circuit. It's experience literally shaping the brain's hardware.
Okay, so the structure gets built and refined. Now, how does the brain maintain a stable view when we start moving, especially our eyes? They jump around constantly, but the world doesn't jump with them.
Right, the smooth world illusion.
Yeah.
Our eyes don't actually scan smoothly most of the time. They make these incredibly fast, jerky movements called saccades.
Little jumps.
Very rapid jumps. This is actually to prevent massive motion blur on the retina. If you tried to pan your eyes slowly across a scene, the image would smear terribly. Saccades give you brief moments of relative stability where the eye takes a snapshot.
And the brain stitches these snapshots together.
Yes. It integrates the information gathered during those brief fixations between saccades to create our perception of a smooth, continuous scan and a stable visual world.
And the reason the world doesn't seem to lurch every time our eyes jump is due to the outflow theory.
That's the classic explanation usually attributed to Helmholtz. It's also called efference copy.
Yeah.
The idea is that when your brain sends a command to your eye muscles to make a saccade, a motor outflow or efference, it sends a copy of that command internally to the visual processing areas.
Like a memo saying, ignore the retinal slip that's about to happen, I ordered that movement.
Exactly. This internal copy of the movement command effectively cancels out the sensory signal coming from the retina that says, the whole world just shifted. The brain predicts the sensory consequences of its own actions and subtracts them. That's why the world looks stable when you move your eyes. But if you gently poke your eyeball, the world seems to jump because there was no motor command, no efference copy to cancel the retinal motion.
That active cancellation is amazing, and it seems like there's a similar kind of active, coordinated process when we track an object moving across the center of our vision, right? When it has to be handed off from one brain hemisphere to the other.
Oh, that interhemispheric handoff research is fascinating. Because your visual field is split, right? Stuff to your left goes initially to your right hemisphere, stuff to your right goes to your left hemisphere. So if you're tracking something smoothly, moving from left to right across the midline.
At some point, the primary responsibility has to switch from the right hemisphere to the left.
Exactly. And the research shows this isn't just a passive thing, like one side drops it and the other picks it up. It's an active, coordinated transfer process.
How did they figure that out? What signals are involved?
They look at brainwave activity, specifically different frequencies, in animals performing this tracking task, focusing on the prefrontal cortex, which is involved in coordinating these things. They saw a distinct pattern of activity orchestrating the handoff.
Like a neural communication protocol.
Almost, yeah. It starts with high-frequency gamma waves, spiking in the hemisphere that's about to hand off the target, the sending hemisphere. Gamma is often linked to active processing and encoding of sensory details.
So packaging up the information.
Kind of like preparing the data packet. Then, crucially, about a quarter of a second before the object actually crosses the midline, alpha waves start to ramp up in both hemispheres.
Alpha. Aren't they usually associated with relaxation or inhibition?
They often are, but here they seem to play a role in timing and perhaps suppressing irrelevant activity or noise during the critical transfer window. Alpha peaks just after the crossing. It's like a, get ready, prepare the channel. Signal acknowledged by both sides,
Clearing the way for the transfer.
plausibly. And then finally, after the crossing is complete, theta waves peak in the hemisphere that just received the target information. Theta is often involved in memory encoding and spatial processing.
So confirming the handoff and integrating the object into the new workspace.
Exactly. It looks like this orchestrated sequence, gamma in the center, alpha ramping up in both. Theta peaking in the receiver ensures a smooth, seamless transfer of the object representation across the midline, maintaining stable tracking. It's like a perfectly timed relay race or cell tower handoff.
It really undermines any simple idea of strictly localized brain function. Everything is so interconnected and dynamically coordinated. Okay, let's zoom out again. We've talked about the mechanisms, but let's look at the bigger picture. Vision optimized for survival, not perfect accuracy. What are the consequences, the predictable flaws this creates?
Well, the main consequence is that biological vision is always a niche solution. It's incredibly well-tuned for the specific problems an organism needs to solve in its typical environment. But that tuning comes at the cost of generality. When the environment changes or when presented with something outside its designed scope, the system can fail, sometimes spectacularly.
The frog example is classic here, right?
Exactly.
Super specialized.
A perfect illustration. Frogs, most species anyway, lack a phobia for high detail, and their visual system is heavily biased towards detecting moving things. Small, dark moving object, probably a bug attack. Large, dark moving object overhead, probably a predator freeze or flee.
But a stationary bug
Is effectively invisible to the frog. Its visual system just isn't tuned to register it. That's a trade-off. Hyper-efficient for detecting prey and predators in motion, completely blind to static food. Extreme optimization for a narrow set of survival-critical tasks.
And then the dragonfly example, that's most tragic, shows how optimization can be fatally tricked by novelty.
It really does. Dragonflies evolved over millennia to find water for laying eggs by detecting the specific way horizontal surfaces polarize light. Water polarizes light horizontally. It was a super reliable cue.
Until we invented asphalt roads.
Exactly. Smooth, dark asphalt roads, especially when wet or under certain lighting, happen to polarize light even more strongly horizontally than natural water does.
A supernormal stimulus.
Precisely. The dragonfly's highly optimized polarization detector gets overstimulated. It screams, "Perfect water, best water ever," and overrides any other conflicting cues. So they lay their eggs on the hot, dry asphalt where they inevitably perish.
A technology asphalt inadvertently creating an evolutionary trap by exploiting a previously adaptive sensory bias.
It's a sobering example of how optimized biological systems can be vulnerable to unforeseen changes in the environment, especially human-made ones. Makes you wonder about our own visual biases.
Absolutely. And speaking of internal states influencing vision, what about imagination? How related is seeing in your mind's eye to actual seeing?
There's actually a remarkable amount of overlap, neurally speaking. When you visually imagine something, picture a beach or a friend's face, brain imaging shows significant activation in many of the same areas that are active during actual visual perception, including regions within the visual cortex itself.
So imagining uses some of the same machinery as seeing.
It really seems to. It supports the idea that perception isn't just a passive intake of data, but an active process of construction, one that can be driven by internal signals, memory, intention, as well as external ones, light hitting the retina.
And are there specific brain networks involved in blending what we see with what we know or imagine about a place?
Yes. Researchers talk about the scene network. This involves key areas like the retrosplenial cortex, RSC, and the occipital place area, OPA. These seem to act as hubs for integrating your current view with your broader spatial memory of the environment.
Helping you build that feeling of being in a coherent 360-degree space, even though you only see a fraction of it at any moment.
Exactly. They help stitch together the current snapshot with your internal map, creating a stable sense of place. This network is likely why memory priming works so well visually. Seeing a related image briefly can speed up recognition because it pre-activates this network's representation of the scene or context.
So our brain is constantly weaving together sight and memory. But despite all this incredibly complex processing, there's still a fundamental speed limit, isn't there? A bottleneck on how fast we can actually recognize things.
Yes, there seems to be a hard limit, a quantifiable constraint. Studies looking at how quickly people can identify sequences of images flash briefly suggest there's a limiting channel capacity for conscious visual recognition.
Like only so much information can get through per second.
Pretty much. The brain can process vast amounts of visual data unconsciously, but the rate at which we can bring that information into conscious awareness and identify what we're seeing seems bottlenecked. The estimates vary, but they often land somewhere around 21 to 28 bits per second.
Which isn't actually that much, considering the richness of the visual world.
It's not. It highlights that our conscious visual experience is likely a highly compressed, summarized version of the total information being processed. It's another reminder that our perception is selective, prioritizing what's deemed most important.
And this biological bottleneck, these limits,
Yeah,
that's where technology really starts to step in, right? Especially in medicine.
absolutely. Medical technology has been augmenting human vision for decades, allowing us to see things far beyond our biological capabilities. Think about the surgical microscope, first used in neurosurgery back in 1957. It utterly transformed the precision possible.
And now we have things like VR and AR in surgery.
Exactly. Virtual and augmented reality are becoming huge. They help with training surgeons, planning complex procedures, and providing real-time visualization during surgery. In neurosurgery, for instance, AR is reportedly used in something like 70% of applications where it's relevant overlaying MRI scans onto the patient's head, highlighting critical structures.
And using special dyes, too.
Yes, like fluorescent dyes that make certain types of brain tumors, like malignant gliomas, glow brightly under specific light sources used with the microscope. It makes the invisible visible, allowing for more complete removal. Technology enhances precision, visualization, data integration, way beyond the naked eye.
Which brings us right to the cutting edge. Artificial sight. Devices designed to replace or bypass parts of the biological system.
This is where the potential is immense, but also where the challenges become really stark.
Let's start with the promise. What are some of the newer approaches, like the Prima device from Stanford?
Prima is a really interesting concept. Instead of larger electrodes stimulating the retina, it uses arrays of tiny, independent photodiodes, basically miniature solar cells implanted under the retina. They're incredibly small, a fraction of a human hair's width.
And they act like replacement photoreceptors.
In a way, they don't use external cameras. Instead, the patient wears special glasses that project patterns of near-infrared light onto the retina. The healthy eye doesn't see infrared, but these photodiodes absorb it and convert it directly into electrical current, which then stimulates the nearby retinal neurons, the bipolar cells, that are still functional, even if the rods and cones are gone.
So it bypasses the damaged photoreceptors using light itself? What are the advantages?
It's modular, potentially allowing for higher resolution by packing more photodiodes in. And because it uses infrared projected onto the damaged central area, patients might still be able to use any remaining peripheral vision they have alongside the artificial central vision. It's a very clever approach.
That sounds incredibly promising. But the sources paint a much darker picture with the story of the Argus II implant. What happened there?
The Argus II. Developed by a company called Second Sight, it was one of the first commercially available retinal implants. And for several hundred blind patients worldwide, it was genuinely life-changing.
What could they see with it?
It wasn't normal vision, more like patterns of light, phosphenes, but it was enough for many to distinguish doorways from walls, locate large objects, sometimes even make out large letters or see bright lights, like Christmas lights. For someone living in total darkness, regaining even that level of perception was profound.
A real miracle for a time.
For a time. But then, in 2020, Second Sight effectively abandoned the technology. They shifted focus, ran into financial trouble, and eventually filed for bankruptcy protection.
Leaving hundreds of patients
Yeah.
with implants in their eyes, unsupported.
Exactly. This is where it becomes a massive ethical issue. Over 350 people globally had these devices implanted. Suddenly, the company supporting them, providing repairs, software updates, replacement parts for the external hardware, was gone.
So if something breaks.
They're potentially left with a non-functional implant. Their artificial vision could just disappear permanently because there's no one left to fix it.
That's devastating. Are there risks to just leaving a defunct implant in place?
There can be. Beyond the immense emotional toll of losing that regained sight, there are medical concerns. The implant has internal and external components. If the internal part, the bit actually in the eye, were to cause inflammation or other complications later on, removing it could require complex and expensive surgery. One patient was quoted tens of thousands of dollars.
And it could interfere with other medical procedures.
Yes, particularly MRI scans. Having a metallic implant could make getting necessary MRIs unsafe or impossible. So patients are left in this terrible limbo, a potentially useless, possibly problematic device embedded in their eye with no support structure.
The critics quoted in the sources argued the company sold these based on desperate hope rather than proven long-term performance. It raises huge questions about responsibility with breakthrough medical tech.
It really does. It highlights a potential gap in regulation and corporate responsibility when it comes to long-term support for these kinds of life-altering implants. Patients invest enormous hope, undergo invasive surgery. They deserve better than being abandoned when a company's business strategy changes.
So what's the proposed solution? How do we protect patients in the future?
One key idea being pushed is mandatory escrow documentation for critical medical devices like this.
Escrow, like for buying a house.
Similar principle. The company would be legally required to deposit all the essential technical information schematics, source code for the software, parts lists, manufacturing and maintenance protocols into a secure, independent third-party escrow account.
And if the company goes bust or drops the product,
That escrowed information is automatically released to a designated entity, maybe a patient advocacy group, a nonprofit, another company willing to take over, support someone who can ensure continued maintenance and care for the patients who depend on the device.
so the knowledge to keep the technology running isn't lost if the original company disappears.
Exactly. It provides a safety net, ensuring vulnerable patients aren't just left stranded. It's about balancing innovation with long-term ethical responsibility.
That escrow detail feels crucial. It bridges that gap between the brilliance of the tech and the reality of supporting patients long term.
Okay. That brings us towards the end of what's been a really wide-ranging, detailed exploration.
It really has spanned the whole system, hasn't it? From the almost unbelievable mechanics of RGC axons adjusting their speed to compensate for distance,
All right, that synchronization is amazing.
to how art and neuroscience both land on the same conclusion about brightness being fundamental for seeing form, with color as this, well, almost luxurious layer on top. And then following the signal into the brain, seeing those classic models like the dual stream hypothesis get challenged by new AI insights.
And tracking how the brain uses those intricate brainwave patterns, the gamma, alpha, theta, to manage that complex handoff of information between hemispheres during motion tracking.
Yeah, the coordination is incredible.
I think the core lesson, synthesizing it all, is that human vision truly is a masterful illusion. But illusion not meaning fake, meaning an active construction.
Built rapidly, adaptively.
Exactly. Prioritizing function, prioritizing survival over perfect, literal fidelity to the raw input. It's an inherently constrained system, optimized for our evolutionary niche.
But a system we're now increasingly able to challenge, augment, and even try to replicate or repair through engineering.
Provided we grapple seriously with the ethical responsibilities that come with that power, as the Argus 6-second story shows. And if we connect this to the bigger picture, you see this fascinating convergence. AI models are getting better at mimicking, sometimes even exceeding, aspects of human visual processing. And neurotechnology is getting better at interfacing directly with the brain's visual hardware. The line between perception and technology, maybe even perception and imagination, is getting blurrier.
Okay, so that leads us to our final provocative thought for you, the listener, to take away.
Consider this. You now know your vision wasn't evolved for absolute accuracy. It was optimized for survival. It takes shortcuts, it makes assumptions, it fills in massive gaps using memory and prediction, all to give you a useful, real-time model of the world.
Good enough to keep you alive.
Right. But now, AI research is starting to create vision models that, in specific ways, might be more robust, less prone to certain illusions or tricks, maybe even more consistently accurate than the biological system evolution gave us.
So the question is,
Does the future of enhanced human sight involve embracing technologies that might make us, in a sense, less biologically human in our seeing, but demonstrably more perceptually accurate? Where's that line, and what does it mean for our experience of reality?
a fascinating question to ponder. What does truly seeing better mean in the age of AI? Lots to think about there.